still at a very early stage and far from assured. I hope that we can continue to work together, the project providing information and CSKT making management choices to attain some measure of security for this gritty and resilient lineage of bighorn. Thank you.
Now we're going to show some videos.
My name's Colin Ruggiero. I'm co-presenting. I'm doing the minor role in the presentation, but I'm a documentary filmmaker from Missoula. And I've been following Jack around for quite a few years now, getting footage of bighorn and also his research in the hopes of making a documentary film about. Jack's research is really pretty groundbreaking, and the longevity of his study is impressive. And so I'm hoping to make a film about that, about bighorn in particular, but again, centered around Jack's research. So I'll just show a couple clips real quick so we stay on time, and then we'll have a chance for the Q&A, it sounds like.
You can use your mouse now.
These are pretty brief, unedited clips. I'll just give a brief intro to one. This first one, they all kind of center around things that are focuses of Jack's and his research, but this is with regard to the mating system. I could say a lot about this, but I don't want to run out of time again, but this is just this last rut. And this kind of shows the first sort of glimmers of a return to a normal bighorn sheep rut at the bison range. These are two 4.5-year-old individuals kind of vying for dominance during the rut. We can unpack that later. There is sound with these video clips. It's not crucial to their working correctly, so we'll just watch them silent and a little stuttery for the moment. This is Jack just expressing that these two individuals have a kind of a rare situation where one doesn't cede to the other for dominance, and they continue to kind of battle it out even after a dominance hierarchy has been established. This next clip just relates to phenology and the timing of bighorn ecology. This is a birth that I filmed a number of years ago. These are so much more exciting with sound. This last one is just a camera trap clip. I've over the years, in coordination with Antoine at the bison range, I've been leaving motion censored cameras out in key areas to get footage that I wouldn't be able to get otherwise with bighorn. So play is a big part of bighorn life, and hopefully the film I'm hoping to make. And so this is just kind of an extended clip of lambs playing, but also a somewhat rare all-age play event on some cliffs they like to use. And you can see the adults kind of get in on the action and run around and play also. Unique to be able to get a camera that close to them. thanks. This was the one clip that just had kind of boring natural sound because the camera's stuck in a plastic box. I think that's our 15 minutes. So we'll just maybe go right to Q&A or hand over to you, Matea. I don't know.
Yeah, are there any questions?
Mint and Sage have an interesting relationship. Normally, what RAMs do is they compete for rank in a hierarchy, which determines.
Go ahead, Tom.
Access, and they don't fight over individual use.
Sure.
If they're
Question, Jack, on the, so it's good news to see that there's a recovery coming. So would it be, in your opinion, would it be okay to augment that herd from sheep outside of the bison range at this time?
I would be confident from the point of view of the bison range bighorn that they're not a threat to the animals you might introduce. But you would want to, I think, test, maybe quarantine the animals that you're bringing in. The other thing that would occur to me, we actually, for this genetic rescue, brought animals in, years ago. And we were really anxious not to just sort of swamp the population with animals that are familiar with a different area and perhaps create movements that weren't there. So we trickled young animals in. And the reason, for doing that, number one, is not swamping the population. So the local culture is the one you retain. But also introduce young animals, 1 and 2 year olds, that haven't established necessarily their traditional, space use. So they're more apt to sort of join and meld in with the local population. So I think it's kind of delicate, especially there's still goats to the south of the refuge and they carry mycoplasma, maybe a little less likely that the bighorn would visit them, but and you never know when another domestic population might, pop up around the refuge. So I think it's kind of important to do it delicately so that you don't introduce new movements that might lead to another epidemic. And so that would be thinking about where you bring animals from. Wild Horse Island is one of the places that we brought animals in earlier on. And the idea was that not only were they young, but they weren't in an area where they were accustomed to long movements. So I guess, there are lots of issues, not just disease in terms of augmenting, but maybe it's not necessary.
Go ahead, Greg.
Yeah, I'm kind of curious over right here. Over several decades, you had moderate fluctuations in the population of the bighorn sheep. And then you had a fairly sharp drop off, introduced, if you will, the genetic rescue. Then you had an unprecedented growth, quickly followed by an unprecedented decline. I'm just wondering, given that timeline where all of the unprecedented things happened within a matter of just a few years of each other, if there was a cause and effect there?
For example, something about having lots of animals led to disease or?
I don't know what it would be, but I guess I'm asking you. But all I'm saying is that decade after decade, you had this going on, and then there was a genetic rescue following another dip. Very sharp increase in population followed by a very, very sharp decrease. That's a lot of events to happen in a very short period of time, given the consistency for the 60, 70 years prior to that. I'm just wondering if the genetic rescue didn't trigger something.
Oh, well, I guess the argument would be that if anything, it protected the population against a disease event of that magnitude. But you know, when I mentioned the sort of this local perfect storm of events, I mean, one of them was the Boone Coles outfit, you know, starting to raise domestic sheep right on the boundary of the refuge. And, you know, there have been episodes of animals leaving the refuge. I don't like to use the word escape because, you know, there's a fence there, but from the bighorn's point of view, that's not necessarily the barrier it is for bison. And bighorn are designed to use the landscape on a regional scale. So we expect bighorn to move around. So they've done that in the past, but typically as a sort of a local exploration by 1 and 2 year olds. But so one thing that happened, and I think it's a coincidence, I don't think it's connected with rescue, is that suddenly there was a large flock of domestic sheep sitting right on, you know, pretty much on across the Jocko, and the highway, but right, and sometimes on this side of the highway, right next to sheep range. So I guess that's the way I look at it. One could say that there was no evidence that there were too many animals, that there was pressure to leave the refuge. On the other hand, if there's a certain per individual probability of taking a foray, exploring, you know, the periphery of what they consider to be their home range, but you have more individuals, then you're more likely to get that kind of a thing happening, even if the per capita inclination to leave is the same. So that could be one thing connected with the increase. But there had to be a flock there, you know, they weren't moving far. So I don't know if that answers your question, but that's a long-winded way of trying.
Go ahead, Tom.
Madam Vice Chair, just Greg, for your edification, the U.S. Fish and Wildlife Service knowingly let bighorn sheep back into the range after they've been outside and exposed to the domestic sheep. And that was a protocol that we would never, we didn't allow. We actually put down any sheep we found outside of a place they wouldn't be, because it's such a terrible thing for them to get exposed and then come back. It's just something you didn't, you know, management of sheep in Montana wasn't something that we would allow. U.S. Fish and Wildlife Service and us argued over that. And consequently, we lost the argument and the sheep population crashed.
Any other questions for Jack? All right. Well, thank you, Jack and Colin, for the presentation.
Thanks so much.
All right. Thank you, Jack and Colin. Our next presenter will be Caroline Bean. She is online, and so let me get her all set up.
Hi there, can you hear me?
Yes. Hi, Caroline. Welcome.
Hi, thank you for having me remote. I'm sorry to not be there in person. I have a family wedding, or else I would be there. Can you all see my screen?
Yes.
Okay, great. All right, well, thank you so much for having me. My name is Caroline Bean. I'm a doctoral student at the University of Montana, and I'm happy to be here presenting on my dissertation research titled A Transforming Terrain, Exploring the Social Dimensions of Managing Ecological Transformation. Before I dive into my presentation today, I want to acknowledge that this work was funded by the Northwest Climate Adaptation Science Center with support from the University of Montana. My doctoral committee members are listed here, and I want to draw a special note of gratitude to Mike Derglow, the climate change coordinator for CSKT, who was a vital thought partner throughout the development of this research. Mike was a part of a group of partners that really helped me develop this research over the course of 2 years. We had representatives from all of the logos here represented on the right, and partners worked really closely with me to inform the study, including the study boundary, the population we worked with, the modeling that we did, as well as the design of a workshop. I'll discuss more in just a moment. And I'm grateful to have used this approach because I think it's been a really solid way of ensuring that the knowledge that was generated from this study can be both of a high quality scientifically, but also relevant to local partners on the ground. I'm going to be using the term ecological transformation quite a bit in the next 10 or 15 minutes. So to first define this term, ecological transformation is a significant and irreversible shift in multiple ecological aspects. So an example of this is how a dry mixed conifer forest, photographed here on the left, may transform permanently into grasslands and shrublands after a high intensity wildfire. And the likelihood of ecological transformation is increasing due to climate change. So in the past, we've assumed that ecosystems have this natural fluctuation within what's called an envelope of variability. But that type of stationarity within that envelope of variability is becoming less likely because of changes in temperature and precipitation due to climate change. And with the increasing likelihood of ecological transformation, that's bringing a new type of complexity and uncertainty to land management. Returning to or restoring historical conditions is becoming increasingly resource intensive. And so while we can understand that change might occur, there's still a great deal of uncertainty about what type of ecological change might result, as well as the probability of success of land management actions. And so into this kind of new challenge to land management was introduced the RAD framework, which was intended to structure future-oriented conservation amidst ecological transformation. It's often depicted as this triangle to outline 3 potential pathways to guide responses to ecological transformation. Resisting change and trying to maintain the current or historical ecosystem structure and function, accepting change and having no intervention and just allowing an emergent ecosystem structure to come about, or finally directing change and having active involvement to try to steer transformation to a new ecosystem that is different from historical conditions. Resist, accept, direct, again, often depicted as this simple triangle, but in reality, the RAD framework exists within this really complex web of a social ecological system where there's these multiple interacting elements and feedback loops, and it's truly an example of the system being more than the sum of its parts. And it's not just ecological systems that are at play within this system. It's thinking about social systems, our culture, politics, technology, and the economy. It stretches across many management boundaries and calls upon many partners. And so given all of this, we can begin to predict that managing ecological transformation will raise really difficult and controversial questions. Engagement is already named as a pressing challenge for land managers without even considering ecological transformation, and the likelihood of change introduces new difficult conversations to have. This might occur around the idea of trade-offs, where something might be gained while another is lost, might occur as choices are made to resist, accept, or direct change. And while there's been excitement over this idea of upstream engagement, which is convening diverse stakeholders and partners well in advance of a decision, we don't really know how that will do yet. There's been very limited social science research on how we navigate ecological transformation, which is really the crux of my project. So I had 3 major research questions guiding my work. The first being, how can diverse stakeholders utilize the RAD framework to provide both structure and flexibility for collaborative approaches? Second, how the personal, our emotions, our spirituality, and values can surface when considering ecological transformation and the RAD framework. And then finally, trying to test how upstream engagement can support decision-making with the RAD framework and how those engagement strategies might be designed. So I'm going to quickly go through my findings for each of these research questions over the next 9 or 10 minutes. go through the methods. So I used a range of methods to try to answer these research questions. I worked with federal, tribal, state, local, and NGO land managers and associated staff, drew upon the experiences of ecologists, as well as representatives from many community groups interested in land management. There were 44 participants. They each participated in 2 interviews and a pilot workshop. The pilot workshop was held in Missoula in October, and we went through a mock management decision-making exercise to test the RAD framework and sort of how it held up. Informing both the interviews and the workshop was some extensive modeling on what types of ecological transformation might occur in the Flathead Reservation, Missoula County, and Lake County. And that modeling and analysis was led by Dr. Kimberly Davis of the Rocky Mountain Research Station. I also used a post-workshop survey to collect some quantitative data in addition to all the qualitative data that was generated from the interviews. And in terms of analysis, interview data was inductively and deductively coded with a tool called NVivo 14, and I also generated descriptive statistics of the post-workshop survey. Moving into results. For that first question about how the framework could provide both structure and flexibility to collaboration, I found first off that managers were very excited about cross-boundary and collaborative efforts as a response to ecological transformation. Folks said that it was, quote, kind of pointless to try to address ecological transformation without cross-boundary collaboration. But at the same time, working across boundaries was dampened by existing organizational constraints. So participants often spoke of their, quote, marching orders that might be associated with their agency's regulations or rules or mandates, which might dictate whether they could resist, accept, or direct change, regardless of whether if they had an opinion on what might be the best approach. And participants saw these as really problematic and limiting factors for having a robust response to ecological transformation. But interestingly, with the RAD framework, participants found, really expressed, and it was played out in the workshop as well, that the framework could provide a foundation for a portfolio approach to cross-boundary work. So because of all this uncertainty around what ecological transformation might look like, participants felt that there was actually a great benefit to pursuing all 3 pathways as a portfolio and creating a mosaic on the landscape, allowing them to build collective knowledge and steward limited resources at this time. And having 3 options also provided this foundation for divergence without opposition. So different agencies could choose different pathways and responses to change without necessarily being in opposition to one another. And it enabled this mixing and matching, essentially, of organizational capacity and mandate with ecological opportunity. So that's the institutional side of things, but I was also really interested in the personal and how emotions and spirituality and values surfaced during these conversations. Spirituality seemed to really inform Indigenous perspectives on ecological transformation and potential responses to it in the interviews that I conducted. Participants spoke about how different landscapes were not just looking different, but spiritually feeling different. They emphasized how important spirituality was to land management responses, as well as emphasized the importance of engaging in spiritual practices to help make a decision about which pathway to move forward with as ecological transformation loomed on the horizon. And there was also a great deal of reference to emotions, to sadness and grief and anxiety that participants felt. Saying that one manager said they felt like they were supporting their staff through something that felt similar to grief. Participants spoke about their kids and what type of future they would inherit and the sadness they felt around that. Spoke extensively around personal losses they felt with the transition of ecosystems such as subalpine forests, using really personal language about how it feels to be in these ecosystems, as well as expressing anxiety over future conditions. There were, albeit some participants who were unbothered by potential ecological transformation, saying things always change, everything is dynamic, and nature will be what it will be. But the predominant response were these references to sadness, grief, and anxiety. And there was also this tension between even making a choice to resist, accept, or direct change, with one participant saying, it should not be up to us to say who lives or dies, what species survive. But then at the same time, there was also a desire to trust what's going on out there, as one participant said. There's a language within the ecosystem that humans don't always hear. We don't always interpret it right. We don't always know what's going on. So an emphasis on humility while facing ecological transformation. So this is a lot to hold in a land management decision. And so that kind of leads into this third question about how can we design engagement processes to support decision-making on the cusp of ecological transformation, and how will we design those engagement strategies? What can we learn in terms of good practices for that? Our workshop, which again occurred far in advance of any decision, we were just talking about hypothetical management responses to potential changes. And we saw that it provoked really critical and imaginative thinking about ecological transformation. There were discussions about the sort of science fiction discussions about hovercrafts and what if the next generation wants to recreate in hovercrafts, and how might that challenge our management decisions of today. One participant said it felt like we were in a group seance for a day. So it was clearly a very immersive experience that was consuming and engaging. And one quote here where a participant said that it forced them to dig deeper and look at the true meanings of things, to think on a broader spectrum, and I really, really enjoyed it. It's been a while since I've been challenged like that. So while there were these positive comments on the workshop itself, and also seemed to provide a good venue for incorporating different perspectives on ecological transformation, with one person saying the workshop could help them in future discussions with agencies who have different values or who are incorporating things like traditional ecological knowledge. And this played out in the quantitative data as well, with participants that were discussing traditional ecological knowledge within the course of their conversations during the workshop were much more likely to agree with the following statements, strongly agree with them, that their group thoughtfully considered the perspectives of all participants, respected one another's knowledge, and respected one another's experience. So while this was really positive remarks on the workshop itself, the RAD framework didn't seem to perform as well. It was more the supporting things at the workshop that were seen as really beneficial. The simplicity of the RAD terminology actually led to a lot of confusion, and the words resist, accept, and direct were used in a myriad of different ways, which led to sort of crosstalk within the groups. People instead were using the words like resisting new information, or accepting that we're not going to get this 100% right, or taking direct action, instead of using these terms in reference to ecological transformation. And that was despite having very active facilitation and this cartoon depicted on screens throughout the workshop throughout the day. As one participant said, we should have used the words octopus, chihuahua, and pineapple instead of resist, accept, direct. So what does this mean for the future of engagement on the RAD framework? Well, it shows that upstream engagement does create room for meaning making about the future. It helps us wrestle with these questions about which times we should care about and whom we should care about in times to come. But it casts doubt on the efficacy of the RAD framework itself. And also, there were power dynamics that manifested in the workshop, with people self-censoring if they felt they lacked expertise. And so finally, to close, some implications. There are several policy and management implications for this work. The first being that a collaborative approach can enable meaningful consideration of all three pathways. And as collective knowledge is built and we are able to experiment, we can shift the feasibility of each pathway and try different approaches to managing ecological transformation. It also suggests the benefits of investing in upstream engagement for more durable management responses, in addition to whatever is already outlined by state and national policy. And then finally, it speaks to the need to legitimize the roles of emotions and spirituality in managing ecological transformation, because they clearly play a large role in these types of decisions and conversations around what our future ecosystems will look like. And with that, I'm excited to answer any questions, and I've got my email right here and happy to share the slides after this as well. Thank you.
Thank you, Caroline. Any questions? I don't know if Mike was in the room, but I'm glad that one of our staff was able to work with you, and it sounds like a really interesting project moving forward.
Thank you. Yes, I know Mike's been engaged on a lot of research with the RAD framework up in Glacier National Park as well, and he's really a leading voice on this, and as well as trying to indigenize it, as he says.
Thank you for spending time with us today.
Thank you so much.
Thank you, Caroline. Next, we are going to call up Rachel Mallison from the Flathead Lake Biological Center.
Hi, Rachel. Looks like you have a couple of our staff with you.
Yes, we're working together. It's great to
Awesome.
have Jasmine and Barry join me up here today. Thank you so much for having me.
Yep.
Pardon me. Do I need to push? No. Oh, ready. Okay. All right. Thank you so much. So today I'm here to talk to you about our Pesticide Stewardship Partnership Program. I'm a professor at the Flathead Lake Biological Station, and we are working together with the CSKT pesticide program and fisheries as well. So I am a freshwater ecologist. I study rivers and streams and a lot of the different things that can influence our aquatic food webs. And there's a lot of things that humans do on the landscape just by living on the land, having homes, farming, treating wastewater, having cities that can result in different contaminants entering into our water. And so one thing that we're really focused on in this program are pesticides. And pesticides are really a broad class of substances designed to prevent, kill, or repel pests. And when we're using the word pesticide, people often will think of just insecticides, but it's really all of the sides that we're talking about. So herbicides, insecticides, fungicides, and more. And they're really used in all sorts of different places: agriculture, forestry, homes, public spaces. And they're designed to be biologically active, so they can affect living organisms. That's what they're supposed to do. But even when they get places they're not supposed to be, they can affect organisms at low concentrations. So we widely use pesticides to protect crops, manage invasive species, maintain landscapes. And there's really thousands of pesticide active ingredients that exist. So this can be a challenge because they don't just stay where we use them. So they're usually used in a particular area, but they can move in the environment, and how they move can depend on how easily they break down, if they bind to soil, how easily they can get into water. As a stream ecologist, we're thinking a lot about how pesticides can influence water and the organisms that live in water. So this figure here is an example. You know, pesticides can be used in that area on the land where that gray box is, but they can run off, they can drift in the wind, they can go down and leach into the soil. So there's a lot of different ways that different pesticides can end up in water, which means they can reach our streams, our rivers, and our lakes. So what is PSPP? This is our new-ish pesticide stewardship partnership program. We're in our third year at the biostation. I'm the principal investigator, the lead on this. I'm Janelle Groff, who's the second person pictured there, is my full-time research coordinator. And then we're also supported by Big Sky Watershed Corps members, and Paige and Callie are both helping this year. And we're really a collaborative network, so we're working together with different groups, from individuals to government agencies, to really help try to improve water quality throughout Western Montana. We work in the upper Columbia River Basin portion of Montana, and that's because we're funded funded by an EPA grant that is active in the Columbia River Basin. And our goal is really to reduce pesticide pollution to waters through best practices and community engagement and education. And we have a lot of different partners, so watershed groups, conservation districts, tribal governments, agencies, landowners, producers, and many more. We're also doing a lot to try and get the word out about our program. So we have a newsletter, we have a website, we have a new Instagram account to try and just let people know about what we're doing to work with water. So what do we do? Monitoring is a big part of our efforts, and we have permission to monitor here on the reservation. So that's why I'm kind of giving you an overview of our broader activities first here. So we monitor surface water, groundwater, and fish tissue, and some other food web components as well. We took over 600 samples last year across many different land use types. We might get up to 750 samples this year. And we use a real GIS-based site selection framework, so we can use GIS layers to understand what the land uses are and then try and divide our samples up amongst those land use types. And then the data will feed into, are entered into the National Water Quality Portal and used to make risk maps. For our education and outreach, we have different workshops, presentations, outreach materials that we're working on, a lot of printed resources, digital resources, and then things like best management practices, native plant guides, pesticide safety, a lot of different types of content that we're creating for public distribution. We also do a lot of community events, conferences, and field days, and have monthly meetings for all our partners that they can attend if they're available. And then implementation. So we have a green stormwater project at Takima Park in Missoula to treat stormwater before it enters the river. That's being almost finished. There's research on pesticide leaching that's being done by a graduate student at Montana State University. We also have native plant installations, working to teach people how they can convert turf grass to native plants. Some funding and supporting different projects on the ground. And then also with the Montana Department of Agriculture, we help fund pesticide waste disposal and container recycling. So there's really a huge array of different activities that we're working on through our PSPP. And those pictures on the right side just show kind of a variety of activities from monitoring to different public events. And there's native plants and just working with the public. So like I mentioned, we're active in the Columbia River Basin portion of Montana. So that's the 12 counties highlighted here in the western part of Montana. And we got to be part of this funding opportunity since we are part of the headwaters of the Columbia River Basin. And we aren't doing all of this on our own by any means. So we have 7 larger sub-award recipients of our core partners that we're working with. I mentioned the Department of Ag already, so we work with them to dispose of pesticides, recycle containers, also with their groundwater protection program. And then Department of Environmental Quality, we're working on education and outreach with them. They're also working to promote native plants and buffer strips going in. The Montana Watershed Coordination Council, we're working on connections and network support, education and outreach. We'll have a watershed tour that's coming up in September for people to come and learn about the program. Montana State University, the different research on leaching, so how are pesticides moving through soils and how might irrigators change how they irrigate. And then the pesticide education program we work with as well. And then what we'll talk a little bit more about today, our partnership with CSKT and our fish tissue and food web analysis. And then I also mentioned that green stormwater project as well. Before we get into the fish, just a little bit more information about what we're finding out from monitoring. So those 600 samples I mentioned from last year, those were all from surface water samples. So we use these glass amber bottles to collect the sample and get them analyzed, mostly at the lab in Bozeman. So for the whole area that we're active, all of these points on the left show places that we sampled last year. The color code shows the predominant land use type. So yellow for agriculture, red for urban, pink for suburban. And then the map on the right shows the locations out of those where we actually got detections of pesticides. So you can see that they are showing up throughout the Columbia River Basin and in the more populated areas. If we look into the results a little bit more, The first thing to note is that the detected concentrations are below the human health and aquatic life benchmarks. But we did detect 39 different analytes, so 39 different pesticides across the entire Upper Columbia River Basin. 34 of those analytes were detected in Flathead County at 38 of the 75 sites that we sampled, and 10 of the analytes were detected in Lake County at 23 of the 60 sites that we sampled. And if you look at those tables on the right, you can see the analytes that were most frequently detected. 2,4-D is detected across the board the most often in Flathead and Lake County and the whole basin in general. And then there's some differences by county, but these are the top pesticides that we are detecting. What are those, you might ask? 4 of the 6 are herbicides, and we're most commonly finding herbicides in our water samples. They can vary from highly water soluble to low to moderately soluble. And what's important to note is that they're being used for crop and non-crop uses. So often people will think about pesticides and think about agriculture being the problem, but we're really finding a lot of detects below urban, suburban, and just general public uses as well. So non-crop uses can be roadsides, rangeland, forestry, yards, all sorts of things. And then we also detected 2 fungicides. So if we look closer to home here on the reservation, we see these points are the sites here more locally. The red points are the sites that we're working with Jasmine and Barry to sample, and the green locations are other water samples that we've sampled. And we are excited to test fish tissue at those sites that I showed in red. Fish in the Columbia River Basin have been found to contain elevated levels of pesticides, including legacy DDT and all sorts of other contaminants in multiple species. These are Jasmine's slides now, so I'm presenting, but she can feel free to hop in and help anywhere here. Did you want to talk about the Yakima Nation?
Sure. So about 10 years ago, it came to our attention, Yakima Nation did a study similar to this, and they were finding their salmon, their clams, their whitefish to be 6 times the health limit. And so that kind of woke us up to wonder, what we were finding here. And also fish that are smoked and dried, for some reason, pesticides concentrate heavier in smoked and dried meat. And so that was like a cultural concern. For tribes, obviously we cook our fish that way a lot. So I just wanted to bring that to council's attention.
So in 2025, we, mostly Barry and his crew, but I get to say we, collected 43 different fish from different habitats. And we're also collecting many more fish in 2026. So the species are listed there on the left: lake trout, rainbow trout, brown trout, northern pike, large and smallmouth bass, brook trout, yellow perch, and northern pike minnow. Some of the tissue is taken from the fish, it's frozen, and then shipped to the lab in Mississippi for analysis. And again, the sites that are being sampled are shown in the red points here. So Flathead Lake, Flathead River, a little bit of Root River, Jocko, Hot Springs Creek, Ronan Spring Creek, and then Crow Hillside, Nine Pipe, and Horty reservoirs. So we're trying to get a number of different habitats and fish from them. And then I'm going to let Jasmine take back over for what we have found so far.
Okay. So we don't have the results yet for 2026, or we'll be sending those fish off soon. But for last year, we mostly found legacy pesticides. So DDT was banned in 1972, but we are still finding the breakdown products of DDT 52 years later. So that's the 4,4-DDE, PCB, those ones are breakdown products of DDT. The 2,4-D and dicamba are currently used pesticides. 2,4-D is kind of used everywhere. Dicamba is controversial. It's been banned twice and reinstated by the Trump administration twice. Dicamba does drift and go everywhere. The company tried installing like drift technology into the chemical itself, but for whatever reason, when it hit the environment, it didn't work how it's supposed to work. So you guys may or may not want to do further regulation on that at some point, but those are the two current ones we're finding. Dicamba, Mississippi lab has it at 2 parts per billion. The health limit for children is 900 parts per billion. So we're far below any health concern, but it is building up in our fish. Yeah. This is important. The legacy pesticides. I have the health benchmarks on the left of 0.5 ppb. They don't have health benchmarks for every single parameter, but for DDT as a whole, it's 0.5. At the levels we're finding it, we found it in various fish, didn't matter if it was lake trout or pike or whatever, 23 to 40 parts per billion, 6.7 to 12. This is 23 times the exceedance of the health benchmark. So this can cause breast cancer, liver cancer, or depending on how many fish people have consumed from the 70s till now, right? Sorry, go ahead, done.
I didn't get to stop.
That's pretty much all. So round 2, go ahead, Barry. When are we sending round 2 in?
That'll be tomorrow. But I would just like to add, it's been very positive working with the biostation on this project, and Rachel's done a great job, and she gathered up quite a lot of money, and that's been very helpful, and Jasmine's been great. And in the fisheries program, it enabled us to do a reservation-wide survey looking for contaminants that we didn't know were there. So far, we haven't found much, but we've essentially sampled our cleaner sites. Samples that will go in tomorrow will be more from the Crow system that's more likely to have contaminants. So at the end of this, we'll have a good survey of where the problems are, and then maybe follow up on zeroing in on that even better.
Thanks so much, Barry. The only other thing to note is that I have a new master's student starting, and we're excited to sample some more of the food web at these sites. So we'll be doing more water samples, algae sediment, and macroinvertebrates as well, to pair with the fish data. And we are just excited to always share about our program in general and get the word out. I think we're out of time, so I'd be happy to take questions, but it's just been really wonderful to have this collaboration together.
Thank you, Rachel. Any questions? Tom?
Thank you, Madam Vice Chair. Well, not necessarily a question, but it's good to see someone who's retired to come back and be here on his own. But Barry Hansen, of course, is retired from our fisheries program. And I just want to congratulate you on your years of service, and what a wonderful program you've made for Flathead Lake, and just a leader in fisheries management. So did you get that captain's license yet?
Thank you, Tom. Still working on that.
And do your domestic sheep play like those bighorn sheep in your cherry orchard?
Couldn't help but think about that. At least I'm not close to the bison range.
Oh, Rachel, thank you for the presentation today. Barry and Jasmine, thanks for being here.
Thank you so much.
Thank you, Rachel and her team. Do we want to take a 10-minute break?
How about 5?
5 minute? Okay, perfect. It's been about 5 minutes. Do you want to get started?
Sure.
Okay. All right, thank you, Mrs. Vice Chair. So our next presenter is Nanette Nelson from the Flathead Lake Biological Station.
Good afternoon. My
Good
name is
afternoon.
Nanette Nelson. Thank you for having us here to talk about a study we actually did. I had to look this up. We did this back in 2021, 2022. But now we've got this work accepted in a journal, and so we're excited to be able to talk about that. The focus of this study centers around CSKT fisheries management decision to restore the native fisheries of bull trout and West Slope cutthroat trout by removing the non-native lake trout adult population. And so we were interested in answering questions on the implications of that removal of the lake trout, namely the flow of methylmercury through the food web in the lake, and also what were some of the health implications for the donation of lake trout fillets and whitefish fillets to local food pantries. So here I have a list. This was work that was a close collaboration with Barry Hansen and the other fisheries people at CSKT, as well as several researchers from the biostation and USGS. And when I was looking at my slides today, I was kind of embarrassed because there's a lot more people that I should have listed here. For instance, Barry's crew at Blue Bay and Native Fish Keepers, they did a ton of work for us. 2 students that were at SKC at the time that conducted the interviews at the food pantries and collected the data that was essential to getting the work done. Brittany Claremont, she also did some work for us. She did a really nice presentation at the cultural committee. And not to mention the Flathead monitoring program that's run out of FLBS and a bunch of scientists at USGS. So I just wanted to give all those people a shout out. And finally, I wanted to recognize the funding we received from EPA from the Columbia River Basin Restoration Program. So the motivation for the CSKT's fishery management program was in part motivated by complying with the Endangered Species Act, but also for the important recovery of critical cultural and subsistence value that are held by many members of the CSKT. The effort behind this program was to remove up to 75% of adult lake trout in order to allow for the native fisheries to respond and increase in abundance. So when we remove that amount of lake trout from the food web is going to change. And the reason for that is they're the top predator. And when you remove the top predator from the food chain, it opens up opportunities for other fish to access resources maybe not available before, and also they have less predation pressure. And because of this change in the food web, we conceived of, or we thought that there might be two possible outcomes from that. One is we expect that the change in the food web would lead to changes in the variation in methylmercury concentration in fish over time. But it also might affect how methylmercury biomagnifies in the food web, the rate at which it biomagnifies. And this has implications for exposure to humans that eat the fish fillets. So this led to two research questions for our project. The first research question really looks at the patterns and controls of methylmercury in the food web in Flathead Lake. And the second one is trying to determine, well, what are these health outcomes that are arising from tribally enrolled women of childbearing age who eat the donated fish at these food pantries. This is not a great image, but I'm not going to spend much time on it. So we decided to come up with this conceptual model to try and illustrate our research design. Basically, the uppermost circle is supposed to represent or has interactions on the ecological level, whereas the lower circle has the interactions on the social, cultural level. And the two intersect at the harvest of the lake trout. Designing this conceptual model helped us to identify key areas where methylmercury is flowing from the lake food web into humans. So in order to keep within time for this talk, I'm only presenting on 3 of our research outcomes. The estimation of the trophic magnification slope, the number of fish meals that minimize exposure to methylmercury using probabilistic modeling, and finally, what are some of the beneficial outcomes that we found by conducting intercept surveys of tribally enrolled women at food pantries. So the first research outcome, I'm going to spend a little time here orienting you on this particular graph. So here we have the food web depicted for Flathead Lake, and it represents, on the x-axis, represents the trophic level as measured by the isotope ratios for nitrogen, which we also can refer to as delta N15. And on the y-axis, we have the log transformed methylmercury concentrations in each of the organisms. The different colors represent different organisms. So the bottom of our food web that we used for this study is depicted in green, and that is Daphnia, which is a zooplankton. And then as we move from left to right on the x-axis, we're moving up in the trophic levels of the food web. And a trophic position is just where an organism feeds relative to other organisms in the food web. So after the Daphnia, we have some myces in pink, the mysis shrimp, as well as chironomids or midges, which are kind of in that mustard yellow, a little bit further off. Next, we have whitefish in blue, bull trout in red, which are a little bit higher on the trophic level. And then finally, the turquoise dots are all the lake trout. And so they are, as I said, they are the top species in the food web for Flathead Lake. And so what a trophic magnification slope is a simple relationship between where an animal is in the food web against its methylmercury concentration. And so we estimated a slope of 0.18, which for other freshwater lakes in North America are average about 0.24. So Flathead Lake is pretty unremarkable in terms of biomagnification of methylmercury. And what what a trophic magnification is simply for, it's the increase in methylmercury concentration in an organism as you move up into the trophic levels of the food web. So what I had mentioned earlier is that we have this measurement as a baseline for the trophic magnification slope for today, or maybe back in 2021, and that this slope might change as the food web changes, as the flathead, as the lake trout are continually removed from the food web. And that slope may get less steep because the remaining fish that are within the lake will be able to access, as I said, resources and also be under less predation pressure, so they'll grow faster. Okay, so research output number 2 was estimating the number of fish meals that minimize exposure to methylmercury from lake trout. And so in this graph, we have lake trout length on the x-axis and the number of times that a particular meal might exceed the federal limitations for methylmercury exposure. And it's dependent on, the exposure to methylmercury is dependent on whether a person is consuming 1 meal of lake trout a week, 2 meals, or 3 meals per week. And a meal is 4 ounces of raw fish, which is about the size of the palm of my hand. And so what we show here is that for lake trout below the length limit for processed lake trout that come out of the native fish keepers out of Blue Bay, that for a person consuming 1 meal of lake trout less than 24 inches or less, there's only a chance, a 3% probability that that meal will exceed the recommended dose of methylmercury. In other words, 3 meals out of 100 meals might exceed that limitation. That probability increases when you start eating 2 and 3 meals a week. So at 2 meals, it's a little over 20%. And at three meals a week, it's more like 66%. So 66 out of 100 meals, you would be exceeding your exposure to methylmercury. We also ran this same analysis for whitefish fillets. And for whitefish, you can eat up to 2 meals per week and only exceed the methylmercury exposure limit 6% of the time. So this analysis here does a couple of things. It confirms CSKT's fish consumption guidance that was published in 2014 in terms of knowing how much methylmercury is in a fish and what fish are safe to eat. But it also adds another dimension to that consumption advisory in that it focuses on the size of the meal, and you don't necessarily need to know how big the original fish was. So now we're on to what are the beneficial health outcomes of the donation of fish fillets to local food pantries. And so we did intercept surveys of tribally enrolled women of childbearing age that visit food pantries, and we asked them questions about their fish consumption and also about their knowledge about the fish consumption advisory. And what we found that 2/3 of this population are eating fish at levels well below the federally recommended exposure. Furthermore, eating of fish is highly beneficial health-wise. And so we feel like those health benefits could be maximized if actually this population was to eat another serving of fish a week and eating a fish that's lower in methylmercury, like the whitefish. So CSKT is providing healthy fish fillets to their community members, especially the most vulnerable ones, which are women of tribal women of childbearing age. But part of that safety is knowing about the exposure to methylmercury. And so here we found that the uptake of the fish consumption advisory was modest among our population. Only about a third of the population that we intercepted at the food pantries knew about the fish consumption advisory. And this is a tough advisory to get across. It's a risk communication that is bounded by benefits on one side and costs on the other. It's been shown that communicating risk communication strategies actually do better in Indigenous populations when it's coming from people that Indigenous people know. And this is reflected in our survey data, and that the number one cited reason for knowing about the fish consumption advisory at all is talking to people they know. And so we think maybe a good pathway forward for expanding the understanding and also the awareness of this fish consumption advisory is to maybe partner with the Food Sovereignty Program and have them act as advisors or ambassadors to safe fish consumption. Oops, wrong way. So here's the title of the pending manuscript that will come out. You can email me, my email's there. If anybody would like a copy, it will have a lot more details on the methods that were used in collecting all the data, plus some of the other findings that I wasn't able to cover today. So with that, I'll take any questions.
Thank you, Nanette. Any questions? Did you say that our food sovereignty program is already working with you, or you're gonna like reach out to them? And
they are not already working with us. They did attend some of meetings that we had, and they were very interested in what we would find out. They didn't want to put anyone into a position that wasn't healthy. And so maybe at some point there would be some partnership between the fish keepers fillet donations and the food sovereignty group. And we could easily provide them with a handout that it describes the fish consumption advisory.
sounds good. Okay, well, appreciate your presentation today. Thank you.
Thank you for having us.
All right, our next presentation is Phil Larson and his team.
Okay, and then I can move it with this. Hi everyone. My name is Philip Larson. I am from all the way from Minnesota, so I came out to visit y'all. Extremely grateful to be here. I'm a professor at Minnesota State University in Mankato, Minnesota, and also on Zoom somewhere, I can't see her, but is my graduate student Jada Rowan, who's a co-presenter of this talk. We focused our research on something a little different. There's a lot of ecology here, but we are geoscientists, and we were looking at the megaflood landforms in the Camas Prairie on the Flathead Reservation, reinvestigating them after sort of years of kind of fame in both the geoscientist community, but also in the public. It's kind of gotten increasingly popular and things like podcasts and YouTube and such. So what we were looking at out here is the famous giant current ripples, or sometimes called mega ripples. So half of the Camas Prairie is on the Flathead Reservation, and the other half, there's a little portion of it, I guess, that's owned by the state of Montana. And so we got a collaboration between them as well to kind of address understanding how these landforms formed. And the cool part about it is they're not just simply giant current ripples, but there's a whole variety of landforms out there that help us understand what I think almost everybody from this part of Montana is familiar with, is the story of Glacial Lake Missoula. And so that's kind of the focus of what we were looking at in our work out here. Before I dive into that, I want to acknowledge our collaborators across the board, including the small army of students that I listed here in bold. We had groups of students learning methods out here, studying this with us over the last 2 field seasons, and funded largely from the National Science Foundation. To get to this point, it actually takes us 1,500 miles away. This work stemmed from a National Science Foundation project in the Lake Superior Basin. And it was because what we found in the Lake Superior Basin were new sites that were emblematic of catastrophic flooding that transformed that basin at the end of the last ice age. And so you can see this Deer Lake spillway here, but also in the red box that just popped up, the Christmas dunes. And the Christmas dunes are these giant mega ripples that were previously unknown in the Lake Superior Basin. And what we've figured out in this work, using the same methods we applied in Camas Prairie, was that these record a large outburst flood event that reshaped the drainage history of the Lake Superior Basin as it became Lake Superior. There were giant large lakes that preceded Lake Superior in that basin. And so that brought us to the Camas Prairie. And the reason why is because we were using this data to try to interpret what had happened, and the famous mega ripples of Camas Prairie provided a great comparative analog for us. And so we wanted to see if we could come out here and look at these to see if our interpretations of things made sense. But one of the problems that we started to find as we went through the literature on what was known about these landforms was that there was actually very little work that had actually been done on these. And they are pretty famous. They are in textbooks that I grew up learning from as I went through college. And so it was kind of surprising. And so we wanted to really dig into these to try to help understand what these landforms are, these really amazingly beautiful places. And so we started with just a pilot sort of comparison. And you can see from the little description here that they are pretty similar features, but there are some differences between the two sites. But they are creating these mega flood landforms, these dune-shaped forms that we see. And we really don't understand how they develop. So we dug in a bit more to try to understand the context, what happened here, and we looked at the story of Glacial Lake Missoula. We've compiled the literature across both the Missoula Basin, but also out into the Channel Scablands of Eastern Washington and through the Columbia River Gorge to try to make sure we understand all of the contextual information to try to help us tell this story. What we know from that story is between 21 and about 13,500 years ago, this large glacial lake filled the basin and drained several times. Some researchers say 42 times, some say up to 100. So there are questions even within that we're hoping to address over the course of our work in the Missoula Basin and in Camas Prairie. Focusing in on Camas Prairie a bit closer, this is a LiDAR digital elevation model that creates a hillshade so we can see the relief in the basin. And what the prior literature has suggested is that drainage through this basin went from east to west through Rainbow Lake Pass, and then eventually got locked in from north to south through Markle Pass and Wills Creek Pass through the southern outlet of Perma Pass. And so we focused on these two yellow X's, the landforms that we found here, to try to be sort of the case study or examples of all of the different landforms, thousands of them out there, to try to understand what happened here. In the prior work that had been done in this basin, they had noted that Glacial Lake Missoula reached an elevation of 4,250 feet. This would have put Camas Prairie under about 1,400 feet of water at the center of the basin, so a massive lake. Around each one of these passes that we were studying, you can look at the elevation of those passes and realize the sort of depths of flows that would have went through them to rip out the gravels and boulders that we now see and create these mega flood landforms downflow from them. They noted a variety of different landforms and mapped them out, as you can see on the right-hand side here. This is a map by Lee in 2009, kind of the most recent study that's ever been done out there, and tried to characterize these landforms in the basin. But what we realized that they were missing was there was no data whatsoever on the internal sedimentary structure of these landforms, which tells us a great deal about how they form, the processes that create them, and the flows necessary to create them. And we also found out that there was absolutely no chronology data. So in other words, how old are these things? When did they actually happen? And so these were things we wanted to address in our work. So we started out by using ground penetrating radar. And this is a shallow geophysical method that allows us to see into these features without actually having to dig them up. So we can do this without disturbing them. And it emits a radio wave into the subsurface and produces something that looks like this. It may look like a bunch of squiggles, or one of my former students said it looks like a bunch of white worms crawling across the map. But what we're actually seeing in there is this structure of the sediments within the landform itself. And so on the red here, we can see Markle Pass, the expansion bar, this large tongue-shaped landform that protrudes out of the pass. And then in the purple box, we see the Wills Creek Pass expansion bar. And here I'm going to highlight some of the things that draw our attention as geoscientists. These red dipping or inclined reflections, and these yellow sort of sub-horizontal reflections that they grade into at the top. In the Wills Creek expansion bar, I want to focus on this yellow box here. We see the same things down there. So we see the same structure in both of these landforms. The stuff above this yellow box are actually giant ripples on top of that expansion bar. So we're actually seeing sort of superimposed or forms on top of other forms, telling us that there were multiple events that formed these landforms in this basin, consistent with the story of multiple drainage events through glacial Lake Missoula's history. Here we see an actual exposure. This is, as far as I know, the only exposure I've ever found in the literature, and I happened to stumble on it driving around out there, which was really cool, at least for me as a geoscience nerd. But here we can see the structure in them, and this is coming at us. So those images you were looking at before were showing flow dipping with the flow direction. Here, this thing is coming right at me. And these red lines are showing us that same structure, those same inclined reflections coming towards us, kind of confirming what we saw in the GPR. So what we think these things are hypothesized to be the result of homopycnal flows, which means that the density of the water coming in is the same of the water it's going into, which makes sense in this context. And then it's a jet of flow carrying sediment with it that is suddenly dropping out and moving stack by stack into the basin over time. And we call these in the geology literature Gilbert deltas. And we think that's what these are, is crude Gilbert deltas. And so we hypothesize that these big tongues, these big expansion bars formed during the earliest, largest magnitude outflows through the basin. Later, smaller flows that may have reached up to Wills Creek Pass then had dunes transported on top of those expansion bars. So we have multiple events forming these landscapes. We also looked at another set of landforms that were mapped previously as what we call anti-dunes, and these anti-dunes would imply that there's really rapid, fast flows across this portion of the landscape. And what we found in the GPR imagery is they absolutely are not anti-dunes. So what we would see is in the direction of flow, we'd see all of these inclined reflections, these light white lines you're seeing going with the flow direction, going the opposite direction, and we don't see that. So in fact, the things that were formerly mapped as anti-dunes are actually just regular dunes, just like the rest of the mega ripples that are out there. And this is really important because when we try to go to reconstruct the flow regimes that created this landscape, we can model this. We're going to be able to then correctly model this. And that's important because prior work, without actually doing the field work necessary to understand this, assumed they were anti-dunes and actually did all that modeling work and got it published. And so we're going to try to correct this and make sense out of this landscape a bit more. We also looked at the giant current ripples farther out in the basin, and we did sort of a long transect through them. And what we can see is that the inclined reflections you're seeing in the GPR, they actually flatten out across space with distance from the spillways. This implies that the velocity decreases as you go out into the basin. And this is what we would expect, which is cool. And we can model this as well. We can fit this into the models to understand the flow regimes that shape these landforms. And so we're basically seeing this in those ripples that are farther out in the basin. We're seeing dunes that are moving into what we call plain bed deposition. Through all of this work, then we have tried to recharacterize the landforms of Camas Prairie, remap them out. And so you can see our work in progress map on the left. This will be part of Ms. Rowan's thesis that will be coming soon. But you can see our new map being created on the LiDAR from this data and the older map on the right for comparison. And the last big thing is that we were actually able to do something called cosmogenic nuclide exposure dating. This basically is a mouthful, but it's a simple way of saying that we can date the boulders when they were exposed at Earth's surface. And so the idea is they're ripped out of the landscape, they're thrown out onto these landforms, and we can take a small sample of rock and figure out when it was first exposed to the atmosphere. The ages that we get from this are shown here in the table, and the locations of these samples are shown here in the map. The ages are all consistent with the age range of glacial Lake Missoula floods, with the exception of those ones in blue here, which are anomalously old, and we don't know exactly what they mean. They could be what we call prior inheritance, that they were exposed previously at the Earth's surface. They were rocks that were out in the mountains that were exposed to the sky and then ripped out by these floods and then redeposited. Or they could be emblematic of some questions that are going on in the literature right now that maybe there was a prior Lake Missoula in an earlier ice age, okay? So now I'm gonna turn it over to Jada. Hopefully she can connect with this.
Hello, you guys hear me?
Yes.
Okay, I'm going to be talking a little bit about linking the geoscience with the cultural history of the landscape. So the core of the Glacial Lake Missoula region lies within the Aboriginal territories of the CSKT, the Flathead Reservation. So they've inhabited the western Montana region since an early period beyond all memory and history, making them among the first to tell the story of Glacial Lake Missoula. So their traditions preserve long-term environmental knowledge and historical memories of this landscape. So the sacred stories, which are traditionally shared in the winter, have been preserved and are still told today to convey the deep cultural, spiritual, and historical significance of the region. And most of these stories center on the coyote who shaped the land by removing people eaters and monsters. And this provides striking similarities about the geologic record of the last ice age, including the advance and retreat of glaciers, ice dam failure, catastrophic flooding under a Great Lake, and climate gradually changing over time. And then this is a map, a DEM map that I created based on a paper I found from Johnson and others in 2014. So this shows Camas Prairie in context of the Flathead Reservation. And then I have some of these indigenous names of the Salish and the Kootenai involved in here from these traditional stories. And I do apologize if some of them are spelled wrong. And then with geoscience technologies, provide tools such as LiDAR, geochronology, and GPR to construct past flood processes and landscape evolution. Meanwhile, we have the stories and indigenous knowledge preserve long-term relationships with these environments. And the integration of these two perspectives allows for a more complete understanding of the history and the landscape through time. And this offers a story of Glacial Lake Missoula's legacy as both a cultural and geologic history reflecting a relationship between the people and the landscape.
So I can jump in here now, Jada. So we still have work to do. So this is best looked at as a progress report because as we've continued to dig into the literature here, we found that there's questions about understanding the ice age advances and retreats over time as well, controversies in the literature about this. So we want to continue our work out here, but would like to do so in collaboration with the CSKT as well. We want to do some of this flow regime modeling and continue our work to really refine this, to tell the story of this landscape from both a Western science perspective and from the perspective of the cultural and oral traditions of the CSKT. And so some of the places that we're looking at this coming summer are noted here. We will be out here working on some state of Montana land, but we plan to share all the data we get from the state of Montana land also with the CSKT when we're done with this work. I will leave it open to any questions now, but I want to first just to give you guys a personal note that this has been, this project in particular has been the most profound of my career. I grew up coming to Western Montana as a kid, so these are some pictures of me back in the early 80s and mid 80s with my family. And now I'm bringing my family to this same place, and now I'm bringing my students, and it has been a remarkable, wonderful experience for all of them. So thank you very much.
Thank you, Phil, and thank you, Jada. Does anybody have any questions? Well, I think it was very interesting, and I good luck with the research that you're doing this summer.
So thank you so much.
Thank you.
Thanks.
All right, thank you, Phil. So our next presenters is Dr. Woodall and her team.
Good afternoon.
Good afternoon. Thank you for having us today. I'm Erica Woodall, and with me is Karen Brown, both of us from the University of Montana. And I wanted to talk a little bit about our longstanding project around pharmacogenomics with the CSKT, and kind of talking about the projects that we've done and some of the things that are upcoming on our radar. I just wanted to give a little bit of an introduction or definition to precision medicine and pharmacogenomics. I think you've heard this from me before, or I think at least some people have, but what this graph shows is that traditionally or usually in medicine, it's kind of a one size fits all. If you get sick and you go to the doctor to be prescribed a medication, everybody usually gets the same drug and the same dose. But what we know is that not everybody responds the same, which is what's showing here is that maybe you have some people who respond, and that's great, that's what we want, but we also might have some people with that medication, they don't respond at all, so maybe they don't have any side effects, but they also aren't getting better. So that's a problem. And then on the other side, we might have people that are having side effects, maybe even very severe ones. So we want to limit that also. So the goal of genetics is to understand maybe why there are these people who either aren't responding or are responding really poorly to medications, and how can we tailor medicines towards people. So maybe it's that those people that didn't respond, maybe if they had a higher than average dose, we would get them into a favorable drug response. And for those people that had the adverse reaction, maybe they're just kind of sensitive and they need a lower dose. Or perhaps they're going to have to, their physicians would have to pick a different drug altogether. So kind of using genetic information to make medicines more safe and more effective. This is a timeline of the partnership with the University of Montana and CSKT. So this project started in 2007 with some initial conversations with Tribal Council and Tribal Health about what a partnership could look like and what kind of research would be of interest to the tribes. Those conversations continued in 2009, we submitted our first big grant that was approved from Tribal Council and Tribal Health that was funded in 2010. So that's really when the work kind of took off in earnest with our first initial funding. In 2010, we also formed our community advisory board that I'll talk about in more detail in the next slide. That was the community advisory board were people that were asked to be on this board from Kevin Howlett and Leanna Muskis. And we've had some turnover over the years, and I'll show you the people that have been involved in that. And just kind of moving along, another thing to point out is it wasn't until 2013 that we published our first work from this. So it took a lot of time to build the trust and build partnership about what we were going to be working on together. And all of our publications have been presented before Tribal Council until the last few publications that have gone to the TRRB. Just following through this in 2020, obviously our research and many things changed. So we had about a 2-year hiatus on our research because of the pandemic, but we've been able to resume the research and finish our aims of the study and kind of thinking about expanded scope that I'll talk about at the end. As I mentioned, our community advisory board, we call it the Community Advisory Pharmacogenetics Council, or CPAC, was established in 2010. And these are just some pictures through the years of various people who've been on the board. The top 3 photos, those are pictures from workshops that we held with our community advisory board around talking about genomics, talking about culture. And on the bottom 3 photos, those are pictures of people coming to my lab so that they could see where the research is conducted. Here's a list of our current and former community advisory board members. So about 20 people over the years have been a part of this advisory board. We're really thankful for everybody's input and help through the years. Vernon is here with us there. He decided he wanted to listen instead of come up. And Marissa is here too. And I also would like to acknowledge the number of names of people who have passed on to and really acknowledge their contributions to this work and thank them. I want to talk about some of the more community engaged research we have done. I'll talk in a minute about more of the biospecimen work, but you can see it really, we have a lot of publications over the years. In this document that I handed out on the last 2 pages, they're in more detail there. They should all be publicly available, but if you have trouble tracking down a publication you're interested, let us know and we can get that to you. But some of the first couple of things was talking about the formation of the partnership, the formation of the community advisory board. We conducted interviews with tribal health providers to hear what their thoughts are on pharmacogenomics and how this could be used in their clinics and in their practice. We've also had several meetings where we've brought stakeholders together to talk about precision medicine research and how we can come together for mutually agreeable partnerships. And then we've written quite a bit on some best practices for researchers who are interested in community-focused research and some of the things that they should be thinking about. This is a few pictures of us at various recruitment events throughout the years. On the top left-hand side, that's my car loaded up from the university with all of the, everything we would need to collect blood samples. So the kind of gray things on either edge, those are portable freezers. They can either be freezer temp or refrigerator temp to keep our biological samples at the appropriate temperature and stable for getting back to campus. We have centrifuges that can spin down the blood that come with us too, and then we can collect blood samples really anywhere. We've had many events where we're recruiting in the conference room just across the hall. The picture in the middle, this was an early study, in this study we were just trying to get the word out. This was a Women for Wellness event, actually so is the one on the top right, recruiting for various studies and getting information out about our research. On the bottom left, this is a picture of us at the Arley Celebration Powwow a number of years ago, where we're recruiting right there, and you can't see it, but the centrifuge is sort of in the corner on the ground, spinning away. A couple more events here on the bottom. One was a back to school event a couple years ago in Polson, and then a former graduate student showing how it's done to collect DNA from a cheek swab. So some of the more biological sample focused research are listed here. Some of the first work was really trying to understand what would be the genetic changes in some of these genes that are really important in how our drugs are processed in the body and how that would then lead to differential drug response. We conducted a study around tamoxifen, which is a drug that's used in breast cancer. This was a study, one of our first ones, that was really important at the time to understand how tamoxifen could be used more effectively for women. One of the brochures that we passed around is a vitamin D study. We were looking at both genetic as well as seasonal predictors. As we'd expect, vitamin D levels would be much higher in summer than winter. We have not published this paper yet, but it has been approved by the TRRB, and we're hopefully going to be submitting that this summer. Antiplatelet study, this was a concurrent study with vitamin D. This is one that is also still in an analysis phase. And then the other brochure around tobacco cessation, pharmacogenomics. This was a survey that we conducted, so it wasn't necessarily biological, but asking members and descendants' perspectives on using pharmacogenomics in aiding tobacco or nicotine cessation. So we got people's kind of feedback on what they would think about a project like that. And this paper was also provided to TRRB and published last year. I kind of alluded to the expanding scope. So in the last few years, we've been thinking a little bit more about not just pharmacogenetics research, but how we can have, what are the considerations for people in rural and tribal communities participating in broadly clinical research, whether that's in partnership with the university or industry-sponsored clinical trials. We've had this Pathway Symposium now 3 times, the most recent being in Polson at the Quatucknuck in October of this last year, which is a picture here. And you may remember we were here last summer talking about a work release for people to attend that symposium. Karen, anything else you'd like to share about this? Okay. This is a paper that we published, just came out a couple months ago, where it was a paper sort of about best practices on working with communities for clinical research. And just wanted to end with this just to show some of the key actions that we summarized in the paper. And the co-authors on this paper were our members of our CPAC. So Vernon and Marissa, Brenda Bodner, Bernie Azure, Patricia Kelly. I think that's it. Yeah. And just a couple of next steps on the horizon. So we've been thinking about funding opportunities to continue this work. There was a letter of intent due for a Robert Wood Johnson Foundation Health Equity Research for Action grant last month. We worked with Vernon Finley on that letter of intent. And then if we are chosen to move forward, that full application will be due in July. And if you have questions, I'll let Karen talk a little bit more about what we're proposing in that grant. One of the things is to help work with tribal health to try to move pharmacogenetics sort of from the research side to actually helping tribal health physicians and pharmacists implement that in the clinic. Another grant that kind of builds on the tobacco cessation work would be to move to more biospecimen sort of study, where we're actually looking to see, does genetic information improve cessation methods for people? So that's a study. We've had a number of conversations with our advisory board and with tribal health about the feasibility of this study. We're targeting this NIH proposal that would be due in October. So that's all I have, just acknowledging a lot of people, both here and in Missoula, that have been part of this work, and also previous collaborators at the University of Washington. Thank you very much.
Thank you. Any questions? Thank you for the presentation. Vernon, you should have been up here. Very interesting topic. Thank you. And we look forward to continue to work with you guys.
Great. Thank you very much.
Thank you, Erica and Karen. So we have one presenter left who is online, Dr. Rose Beardonwalk. Let's check to see if she's here. No. So I guess she's not here. I've reached out, so I guess we're done for the day. And thank you all, council members and everyone who attended today. We will be back in the fall, winter time. Thank you.
Matea, thank you for getting this set up and scheduling it and bringing everybody forward. Again, everybody that presented, thank you for the information today. We really appreciate it.